Welcome, everyone. Thank you for joining me this afternoon for our webinar series, The Next Generation of Scientists. My goal today is to introduce and share content with you that you can use for your students, and we'll be focusing on life science for elementary. Let's just jump right into our agenda then for the day. So the first thing I want to do is kind of cover what we're going to be doing. I always like to do that to start out. There's going to be essentially two different kind of segments. The first segment is going to be about plants, kind of more like the anatomy, soil, seeds, etc. I want to share some kind of great projects students can actively engage with, as well as learn about kind of biology and other aspects of life science. Now, keep in mind, you may or may not have already done some of these activities, but I figured you could glean some new information or mix or match what you hear throughout this webinar. The next segment that we're going to be doing is going to be focusing on animals and insects. So once you're outside, you can kind of visibly see what's out maybe in like a garden space, or if you start digging up dirt, there might be some visible traces of animals or insects the students might see as well. There's some great enrichment and extension activities or even core activities you can use with animals and insects that will also be covered today. So, and then of course, at the very end, we'll have plenty of time for the Q&A. So during this, webinar, I'm going to be focusing on some things that you can use at your school or near where you teach. In this case, I'll be sharing some instances related to school gardens or how to manage plants in the classroom. I'm also going to get into maybe some specifics. I'm not going to. So I'm not going to get into specifics of how to start or manage a school garden because I think that's a more of an elaborate session all by itself. But I'll be getting to specifics about, different strategies and concepts you can teach with one. I like to be kind of clear about that, so I'm not necessarily disappointing anyone by the end or their expectations. So I've kind of written down a list of some of the topics we'll be going into more detail on, and maybe you've heard or seen or used some of these before again, but that's okay. You can share what you've done as well as cover each of these areas. You can kind of share with the group. We've also kind of heard some different terms like, this is referred to as like our outdoor classroom or our outdoor lab. That's typically used by educators whenever they refer to like gardens or plants growing out in these specific spaces in the campuses. So with that in mind, you first might ask yourself, okay, where do I start? Of course, this is dependent on your students, age, capabilities, and your environment, like your resources or budget available. But let's concentrate on the content. Do you focus on parts of plants? Do you focus on the process of plant growth in the soil? Or do you study the system as a whole? Does each activity scaffold to the eventual synthesis of information about the parts of plants and how they will work together? Is everything tied to a calendar of seasons and holidays? You really need to figure out the bigger picture first and then kind of work into the tactics of implementing that goal. So what are student outcomes you want out of this projects and experiments? How are you planning or assessing student knowledge and growth? This will kind of help guide how long and which activities you'll be using with the garden or plant studies. What I'm going to be doing today is give you some different ideas into building that framework so you can kind of run a couple of weeks or a specific unit with some scientific phenomena. So the first thing I'm going to be focusing on, the first topic is chromatography. And you might, yes, there is something shared on the screen. I just want to make sure everybody can see my screen okay. All right, can everybody see my screen okay? I just want to make sure that everybody can see that okay. So the first thing that I'm going to be focusing again on is, again, chromatography. We first talked about, of course, planning the projects, where do I start, and then the different topics that we have here. So just making sure everybody can see my screen okay. I know the last session I ran kind of had a little bit of a difficulty with that. So again, there's our topics that we're going to be covering, talking about planning, where do I start, and then getting into, again, our first topic, chromatography. And you might first think to yourself, okay, wait, what is chromatography? And that's perfectly okay, but I'll explain in simple terms. Chromatography is a method of separating the components of a mixture. And you may or may not have seen or performed an experiment like this during your education. One of the most common lab examples of chromatography being used is with ink. And so right there, I've pulled up a picture. Maybe you've seen something like that image before. If not, that's okay. But essentially, the ink is being separated into different colors or components to allow you to kind of identify all the different substances that make up the ink. This method could be used in forensics, biology, chemistry, food, etc. And there's lots of reasons to figure out the composition of mixtures. So the piece of paper you see was set in kind of like a small amount of solvent, which is the thing that kind of is responsible for separating the mixtures. And I kind of have a little line there to kind of help you with that. So as the solvent rises through the paper, and that's where it kind of starts with that red line, it meets the sample mixture, which then begins to travel up the paper with the solvent. So if you're wondering what materials are needed for this type of experiment, it can be done with some very, very simple materials. Chromatography paper or coffee filters work too if cut into strips. Some sort of container, usually a beaker or a test tube. You probably want something that is transparent, and also something that's easily used for your students to be able to record data in real time. So then you need some kind of solvent and mixture that could be separated. You might be thinking, well, what kind of solvent should I use? And that's a common question. Don't worry. The solvent depends on the mixture you're trying to separate. So commonly used solvents in the classroom is things that are like water, especially if using for younger students and they want them to be hands-on. But with assistance, you can also use isopropyl alcohol, ethyl alcohol, acetone, petroleum either. But before doing the lab with your students, you want to probably experiment with various solvents to see what works best for you. Water could probably work okay with plants, same thing with rubbing alcohol. But you also want to make sure you're comfortable handling the solvent you're using. The next phase is, okay, now that I kind of understand what chromatography is, how does this work with plants? And it really has to do with the leaf color, the green pigment. Chromatography is used to separate the pigments found in leaves. So as you can see, a green green pigment leaf can be easily separated. It shows some different parts of chlorophyll, the xanthophyll and the carotene. And the garden of your school might be surrounded with like trees or shrubs of all types. These are great for chromatography experiments. And if you have a variety, you can compare results. If you're struggling with something to go for or struggling with something to use at your school, you can also use fresh spinach from the grocery store. What's really great about this experiment at the elementary level is that as students get older, they can begin to record data about the different colors and the value of each component within the mixture using the same experiment. So if you see the arrow on the screen on the right there, this is where the solvent began, and you can see it kind of travel up. The students would measure from the dot to the end of each color and record the value. Now, I won't get into technical aspects of this because I don't think it's relevant for necessarily the elementary age yet, but this introduction activity is also great for elementary because now students can visually see and begin to understand the green leaf color, which is the chlorophyll. So what do you do with it? The next time you want to explore leaf color, in the fall or the leaves of your plants in the garden, try this experiment out instead of maybe the traditional collected leaves and read books about why leaves change color. The students will kind of appreciate that change and the hands-on approach to kind of see these more or less quote unquote hidden colors. When you collect the leaves, you just want to make sure they are from the same species. 3 of the same type is typically the way to go. And you can have students match them up to the same type of leaf to kind of confirm that they are the same. Then you're going to kind of rip up the leaves into a container. You're going to use the coffee filter strips or the chromatography paper, and then you can use rubbing alcohol over the course of several days to kind of see that progress. And what you see to the right is more or less what you should see as the end result of that project. And then, of course, think of safety. If you're using something other than water, you can still use this experiment with like kindergartens, but you probably want to have control over the container to ensure your students are safe. You can also use, time-lapse videos with this over the course of several days if you want to help that, like if you're at home or something to that nature. That's also an option and also very impactful as well. As an extension activity, you can also understand why kind of chlorophyll is green. So essentially, it has to do with the leaf absorbs light. As you know, the rainbow shows a spectrum of light. Each color has a wavelength, and the plant absorbs also reflects wavelengths of light. It's just a variety of this, right? So in this case, chlorophyll needs the light wavelengths of blue and red, but does not need green light, which is reflected. And this also ties into why we see different colors, but that can be also explored by testing out which color. Is it, which color works best for the light? So we actually have here a product from Nasco, in case you're also interested. It talks about, LEDs being used for different light wavelengths, and you can test which one best helps plants grow. So it's just another exploration activity if you want to kind of further strengthen students' knowledge about plants and chlorophyll. It's all about the scientific process. We have the product number there in case you hear, but you can see that darker picture with the redness there. That's just showing a red spectrum, but you can change it to yellow, blue, green, etc. And then also measure the student data as well. So if you are interested in something like that, that is an option from NASCO in case you are curious. Dichomous Key. So this is more or less a reference tool that allows someone to determine the identity of items in the natural world. It can be trees, it can be animals, it can be mammals, etc. The key basically refers to the series of choices that lead the user to the correct name of the given item. So what you see here on the board is might seem a little bit of chaos, but what they used was a template, right, of this one monster. And it could be a template from like a coloring book. It could be a plant in this case. It could be monsters, etc. And what you do is you can choose a series of colors and have this. They can take this home as a homework assignment, and they can choose their colors. And so in this case, they used the blue, the green, the yellow, and the red. They use 4 colors, but if you want to make it a little bit more, a little bit less, you can certainly do that. But you can see at the very top there, it says monsters. And then there's a solid body color, or there's a non-solid body color, right? So the ones that are solid are off to the left, the ones that are not are off to the right. And then you can start to see that they kind of break them down into each individual area. So based on this key, you could identify each one of these monsters. And so this is just a great way to talk about classification and how do we actually classify plants and other different objects in the world around us. It's just a really good activity for that. If you want to go a little bit further, you can use this as like give them a scientific name. So if you want to make your own plant after studying the plant units, what would give a plant characteristics? How would it, gather light, or how would it actually grow? How would it get the essential nutrients it needed? So there's a lot of different applications they can think of that you can use as like a summative project at the end. But this is just a one way that you can use for the dichotomous keys. And again, this is for animals, or it could be for plants, etc. Dichotomous keys are used all over the place. So this is just another way you can incorporate with your plant unit, is using a dichotomous key. Okay. Then you can get into the seed investigation. So typically, whenever you use the seeds, you can start off even before you even grow your plants. You can start to use this chart that you see there that, hey, we have a seed here. What is the shape of it? Is it spherical? Is it oblong? Is it irregular? And with the younger students, what you can do is you can actually draw an actual design of what is an oblong shape look like, or what does a spherical shape look like. And typically when I give them this kind of table, I wouldn't even give them the color, shape, size, or specific characteristics. I might just give them the seeds and say, okay, these are the name of the different seeds. Which one of the seeds do you think match up with the name? It's just a really good way to kind of get them to start to think about that, and think about the seeds and where it actually begins. And then after they do that, then you can say, okay, now I'll give you the color, shape, size, and specific characteristics. Now can you match up the seeds to the correct name of the plant? So that's just another way that you can utilize this kind of investigation. And then as they're growing plants from seed throughout the school year, they can start indoors and move to the outdoors. You can grow them in a tent inside. You can put them by the windowsill. If you do put them by the windowsill, you want to be, of course, careful of different drafts and things of that nature. That's why sometimes a growing tent is nice. But then you can transplant them as they grow bigger into clay pots. And then as they get into clay pots, then you can start to put them outside. Usually, like I'm in Wisconsin, typically by maybe the end of April might be a good time to actually do that for the colder weather areas. But that's just one way that you can do the seed investigation from the very beginning and then get into the actually growing aspect. Also along the way too is you can actually create like maybe like a 5-page book, and each page kind of has like a different question that encourages students to integrate their understanding of what they have done that day in the class, along with like the knowledge of the plants. Like, hey, what did we plant? How did we plant the seeds? What do seeds need to grow? What did your pot look like at first, and what do you need see in the pot now? What did you learn? And so you can kind of use this as an assessment tool as well, and you can get artistic too if you want them to actually draw or diagram everything they see, or make a prediction of the different phases, right? So there's a lot of different ways that you can implement this. You can also talk about integrating this with like a Thanksgiving unit, talking about more or less the quote unquote, sometimes referred to as the three sisters, the essential Native American plants, the corns, beans, and squash. Now, those in himself have enough nutrition and vitamins to sustain somebody, but that's why they utilize those different plants. And so you can actually integrate those different plants and that knowledge into your seed unit, right? And saying, okay, this is what we're going to start off with. By the time they grow, did you anticipate that this was going to be, and why would we use these different plants, etc. So it's just a great way to observe both the seeds and pictures of the plants and how they produce. It's just a really good tie-in for everything you have. And so for grades K-2, maybe they are observing the seeds, match them to the fruits that that or plants that are coming to, discovering the seeds need water and warmth to germinate. And then for the 3 to 5 grades, students not only match the seeds with their corresponding fruits and vegetables, but then they also use the Dichomous Key, and then design investigations to answer questions as they generate about the seeds. So you can kind of gauge the different level of activities depending on the level of the student as well. The next thing that we're going to get into is the kind of extension to the seed investigation. So talking about some different phenomena that is happening, for example, plant mazes. So maybe you've seen this, maybe you haven't, but essentially what we have there are plants. There's little bean plants that we put in these shoe boxes, right? And you can put these barriers in the way of where the plant would need to grow towards the light. So at the top there, you have a hole in the shoebox. You close up the shoebox so it's completely dark in there. The plant is well watered prior to closing it over a course of like maybe 4 to 5 days. Then you open up the shoebox and see what is happening with the plant. Well, you can see that these mazes are actually, the plants are actually growing around, and this kind of also relates to tropism, right? So as plants get into to like say for example, you put it on the windowsill, you know that plants are going to lean towards the light. They're going to naturally gravitate. So they're going to point to that particular direction in response to the sunlight. And so that's where this kind of plant is kind of growing. It just kind of shows that even though there's barriers that exist, plants do have some fighting tactics to still get what they need, the nutrients that they need to be able to grow. Same thing for fall weather. If you have leaves that fall, plant growth underneath, when you first maybe rake those leaves when it comes to the springtime, you might see plants that are white. And this is because they don't have that pigmentation yet. They haven't yet absorbed the light from the sun until they turn actually green with the chlorophyll. So just a great way to kind of actually extend that. You can also use jelly beans for the Dichomous Key. This is another great extension activity, talking about if you want to talk about seeds, you can start off with like jelly beans. Sometimes this is a fun activity because then you can actually eat the food afterwards. So it's kind of another engagement piece. But of course, you have to watch out for food allergies, things of that nature. But you have a table, you basically have a key there. That's what that pink piece of paper is. And it kind of walks them through saying, okay, is it a particular color? If it's, green, yellow, or purple, go to line 5, or if it's yellow, go to line 18. And it kind of walks them through those different steps. And what I'll do is I'll actually send a resource that kind of gives you a little bit of a Dichomous Key for jelly beans if you're interested in that as well. Um, but but yeah, so this is just a great way, again, to reiterate to the spectrum of light that works best with plants. So if you do want to investigate with the spectral light, you can certainly do that as an extension to this activity as well. Parts of plants and different investigations here. So what we have here is on the left-hand side in figure 1, this is a plant that's a daisy, a white daisy that's been cut onto a colored water. And so when we think of stems, what is the purpose behind them? We can use these investigations to show that, yes, the stem is like a straw that kind of drinks up the water or absorbs the water for the plant. And we can see that water then extend to the plant itself. So this is just another great visual activity that you can use to talk about the functions of the stem. In figure 2, what the students are showing actually is and I think I missed the picture on here, or unless it's on the next slide. But essentially what you have there is a jar, a student showing a jar and the different layers of soil. So what you can do in this investigation, talking about soil layer, is that you put water or soil in a container, and this is a transparent container again that you can see through, and you fill it up with water. So the soil could be maybe halfway to 3/4. You fill it up to at least that line with water, if not a little bit more. And then eventually over time, you're going to see the different layers change, right? You're going to see the rocks, you're going to see the clay, you're going to see all the different layers that you would need from the plant, okay? And so it's just a really good way visually to represent the different soil layers. And then we can too extend this into, okay, why do we color it brown, or why do we color it red? Why do we color it yellow? Do we color the rock formations purple? Why or why not, right? And so when we talk about art and true facts versus artistic imagination, you can combine that STEAM element too, as talking about, yeah, scientists, they use facts, and when they do this, we want to stay true to the color that we're utilizing. We don't want to just pick a color at random. So that's a great extension activity there. And then, of course, the figure 3, the traditional, hey, let's build a plant poster at the very end after we talk about the plant's functions. We talked about the stem, the soil, the leaves, the chlorophyll, all that together, and you kind of use that as your last project of the case. I think on the next page, yep, so you can see on the right-hand side there, that's where you can see the actual soil begins to separate. It's just a really good example of the different layers for students as a visual representation, because otherwise students might think, well, it's just dirt, it's all brown, right? So that's just a great activity that you can utilize. On the left-hand side, what we used is a old CD case, and so if you can take out that black insert, it kind of does a really good job of keeping a layer for you to be able to utilize with plants and kind of see the root system. So if you want to start there, that's just another extension that you can do. And you can use that little side that at the top there, that's kind of the connection piece where it kind of folds over. You could use that as a watering area, right? And so then you put the beans seed in there, and then it can begin to grow over time. And so this is, again, another way that you can utilize plants in case you don't have a garden other ways. These are just different strategies that you can utilize to talk about the different parts and the needs of plants in your investigation. All right. Now moving on to more or less our second segment here. So we talked a little bit about plants. We talked a little about the functions and what you can do with those, but what's in the soil, right? What are things that might they see around the playground or outside around the school? These are one of those things that you might see. They're known as roly-polies or pill bugs. So these guys kind of roll up into a ball. They're very tiny. A lot of people think of them as pests, but they're really not. They're actually very, very good for the soil. And so typically they're a good indicator that your soil is rich in nutrients that it needs. So that's why you'll see them. They also like cool and damp, dark places. So you might see them under logs or under concrete bricks or other things like that nature. Very good to have and very easy to handle. What's really neat about them, they're actually crustaceans that have kind of evolved. On the bottom right-hand side, you can see an aquatic cell bug. That is basically the water version of that. It's pill bug. But yeah, the pill bugs do have gills, which is kind of surprising. A lot of people think like, wait a minute, they're land animals, right? So why do they have gills? Um, and so these are great for decomposition. So if you have a log that you want to decompose within your classroom and see that over the course of a year, these guys would be great for that. Just one of the different animals that you could utilize with that. And you can purchase these in small units, or you can have the scavenge for your students to scavenge and find these. These guys also like to eat like potato slices. So if you have a slice of potato, those are also really good to use, but really, really great to also incorporate with any kind of plant investigation and gardening, because decomposers are essential to part of this. I also threw in these guys, and you might think to yourself, wow, you're kind of crazy. Why would you ever consider hissing cockroaches? These guys are called Madagascar hissing cockroaches. And they can quite get large. They can get anywhere from 4 to 5 inches long. On the top there, you'll see a male. You can see kind of like those. You can see where the head is. Well, right above the head, you see those kind of two little bumps, prenatal bumps. That's the indicator that that is what a male looks like. On the bottom is a female, and you can see that those little humps are not visible. So that's a distinction between the male and the female. You can also have these are very docile, meaning that they're not going to cause havoc in your room. You can keep them at a cooler temperature so they won't even reproduce. A lot of people are like, well, if they get out, they're going to reproduce and there's going to be havoc and everything else. No, they're actually not pests. They don't reproduce really in cool spaces like in schools and things like that. They're actually normal environments, outside environment. So that's pretty traditional. It's not to say that they can't reproduce, but it's just saying that typically these guys are not considered pests because that's not their normal environment. What's also neat about them is that they create this more or less this hissing sound. As like most predators, they want it to off predators away from them. So what they do is they create this hissing sound to warn people, but it really is just them blowing air through their spiracles, which is found on their abdomen. That's what the hiss sound. So it's kind of a really cool instance. You can do some YouTube videos just to kind of see a little bit more about them if you wish to, but these are really great because then they can also talk about the life science of insects, and also it's an engagement component. So every time they come to your classroom, they're going to be like, oh my gosh, did you hear that, Mr. Nelson has cockroaches in class? No way, really? It just really captivates, and you're going to be known as that class that students want to go to because you have something that others don't, right? And so it's just becoming knowledgeable of understanding how to maintain them. And it's really easy using like Cheerios. You can use egg cartons to put them as their homes. And you can also do a little bit of water in there with like maybe like a cotton ball to help them absorb their water. So it's just a really easy way for you to utilize. You could use petroleum jelly around the top of the cages, so it helps prevent them from quote unquote escaping. Now, I do have to pre-warn you, if the females are pregnant when they're sent to you, they could deliver. So that could happen. Typically, they don't reproduce when it's cooler, but they can, if they're already pregnant, they would actually deliver. And those guys are really, really tiny. So they can get out of those little tiny holes that you would have at the top of a normal screen. But if you want more details and you want to really introduce and give this a shot, I definitely recommend it. And I can help with the implementation of these wonderful animals and insects. So, and another thing too is try to avoid of creepy crawlers or terminology of critters or things of that nature. that's usually a turnoff to students, or you giving them the verbiage of like, oh, that's gross. Well, it's not gross if you really understand the why or reasoning behind it. They're actually not very dirty animals. They actually are very cleanly, and it's really easy to maintain. So just like any terminology you use in the science classroom, it's important to use the correct ones. The next one is mealworms. So these guys are great because oftentimes when they hear the word, students hear the word mealworms, they think, oh, that's a worm. Well, no, it actually is a beetle. And you could use this as a life cycle throughout with all your students. The beetles have wings, but it's kind of like the soft exterior wings. So it's kind of like fused. So it's kind of like a useless wing. So they just kind of walk around. They like the darkness. They're nocturnal. The different stages are kind of interesting for students to kind of see and investigate because it's very unique. It almost looks alien like, especially like the pupa. But it kind of gives the students the understanding that, hey, there's different life cycles out there. There's different opportunities for you to kind of explore the environment around you. These guys are great to learn about, structure and function, the animal behavior, metamorphosis, if you want an alternative to using butterflies. So this is another great way to introduce something that is kind of unique. And you can use literacy as well with this, right? So if you have an investigation, you can create a book and story about this, about the misunderstood mealworm, right? And so there's just a great way that you can kind of segue into using these kind of animals for your different classroom. So some things to kind of consider with animals, because this is sometimes one of those touchy areas. They can be great for classrooms. It adds a great engagement. Again, you're going to kind of more or less be that class that students want to be a part of because you have something that others don't. And if you want to, students can help maintain this. This is a great way for them to kind of get involved with this. It's not for everybody, and that's okay. Just like anything that we learn at school, right? So we teach chemistry, we teach biology, we teach all these different sciences, but not everybody's going to be a biologist, not everybody's going to be a chemist, right? But it's good to expose them to those opportunities because then it gives them that experience to better their understanding of science, but also just become more engrossed into the scientific realm, the scientific community, saying that can be fun, this can be engaging, and there's many different aspects of science, not just this. Choice chambers out the right-hand side, those are great for studying insects and animals, for doing studies, just behaviors and environments, things of that nature. I like to always point those out. You can also use those for plants. So just like I was talking about those mazes, if you want to, you can cover up the bottom piece and keep it flipped up and see if it actually goes through that hole or not. So that's just another opportunity to do that if you want to build a maze within there. If you're also looking to adopt these, I would definitely have a disposal plan in place if you're done with them before you order. It's essentially you do it before you order because oftentimes at the very end of the school year, sometimes if you don't have an idea, it's usually you taking them home. So unless you're going to give it to a student that you know is responsible and is going to do it in an appropriate manner, that's something to be aware of. Oftentimes, teachers will use freezers because they will slowly become inactive, and it's probably more or less the humane way to dispose of them. But again, that's just another way to think of it. You don't necessarily want to give them into the environment because if you're not familiar with what you have in your current environment, it might upset the balance. Now, granted, if it's pill bugs, one or two, it's not going to hurt, I don't think. But it's going to be one of those things you want to be cautious of that because there could be fines and penalties for doing something you're not supposed to do. Again, routine for students, keeping cleaning, feeding, help out with that. You can have a shared set of animals or insects that you use that you can put on a cart, that's a mobile cart, that you can send to one classroom to a next. There's those capabilities. Definitely want to be safe because, of course, those students are quote unquote hanging out by the animals or insects or even by plants all the time. You just want to make sure that they're not mishandling them. And keep things where you know that it's going to be the best impact for your students. So just another way of things to consider when you have animals or insects within the environment. So, and that's kind of more or less the end for what I was going to be showing today. Again, this is the first step. I definitely have a lot more different activities and different extensions if you're looking for them, whether it comes to plants and or insects and animals. But I definitely want to open it up now for some different questions that you might have as far as implementation, or if you wanted to something specific about the activities that was shown here. As you're trying to answer some questions or write some different questions down, I did want to reiterate that there will be a survey after this webinar. Please make sure to take the time to fill that out. If you're interested in some other different activities, extensions, definitely reach out or share something with me if you want to help with planning a unit for yourself. I'm more than willing to do that. My contact information is engage@nascoeducation.com. And you can see that right there on your screen. So again, if there's something that you saw too, you will receive a recording of this. So if you want to do any kind of follow-up with that, that will be in your inbox about 1 to 2 hours after this webinar. Make sure to check your spam folder for that as well. That could be something that you can look to as well. So I'm going to look over here into the questions tab to see if I have anything right there. I currently do not, and you're welcome for the jelly bean activity. Yep, absolutely. I will make sure to get that to you guys as well. I'm gonna actually share some other different files too as well that I have throughout this presentation afterwards as a follow-up. If you are watching this as a recording, make sure to email me at engage@nascoeducation.com to get those resources. That way I can be able to know exactly who needs what. And here, let me grab okay, so I just attached a lab sheet. That is the lab sheet I use for the Dichomous Key. If you want something like that to be able to utilize for your classroom, let me look for all right, so this is the Dichomous Key. I have that on there. So that is also another great activity of talking about the jelly bean activity if you want to be able to use something like that in the handout section. And I also have, I also recorded the blank seed experiments as well. So that also has a drawings too on there as well with some of the seeds examples, just in case you're interested in some of those templates too. So not seeing any questions, and that's okay. Like I said, I'm available via email for some of these different activities and some of the things that you have that you saw there. But again, I can also elaborate a little bit on some of these projects that you can utilize with your plants or your investigations too. So I hope you enjoyed it. I hope you were able to gain or glean some more information on some different exercises. There are going to be future webinars with the next generation scientists, grades from K to 5, middle school, high school, etc. This just happens to be the kickoff for it, and we'll use your feedback to continue to improve these. So I don't see any questions. So thanks much for joining me today, and I hope you enjoy the rest of your day and have a great weekend. Take care, everybody.